on the Luria-Delbruck section. So, you know, I've taught it before, but this time I managed to simplify the calculation. So now the notes reflect what we did in class yesterday, which is, I think, a better way to do the calculation. So I guess the best way to learn is to teach. The second best way is to do the exercises. So I'm glad that some of you are trying the problems I gave yesterday. And if you get stuck, then feel free to reach out to me after class or later today, and I can help you with that. Because, you know, active learning is much more effective than just listening. And that's why I like all your questions. So keep the questions coming, please. Okay, and if you want another problem, just reflecting on what we did yesterday, so the main calculation we did yesterday was about fixation probability. So a mutant just arose out of a population. It has some fitness advantage, maybe not. What's the chance that it overtakes a population? We saw that there are two regimes. One regime where the mutation is neutral, and then the result ends up being dependent on the population size and simply 1 over n, which was sort of obvious a priori. And then the less intuitive regime was where it had a fitness advantage. Let's say it's fitter by epsilon over the others, and the main result we had was that the fixation probability in this case was simply equal to epsilon, or approximately equal to epsilon, and independent of n. So in one case, it was dependent on n and equal to 1 over n, so that's the neutral case. And in the case where there is selection, and one is fitter than the other, then even for a large population where neutral would say there is 0 chance of fixation, here there is some finite chance of fixation that was independent of the population size. And to solve this problem, we essentially viewed it as a first passage type problem where we are looking at this ladder of states between 0 and n, and at every stage we can go up or down in this ladder. But going up and going down had specific forms that depended on the fitness. It wasn't so simple. And we managed to set up a recursive equation for the fixation probability starting from state I. This was this variable's xi. And by setting up a self-consistent equation for the xi, we could solve the problem and find an explicit, quite simple formula for the xi. So if you want to test your understanding on a simpler problem, but the logic is the same, there is a famous problem called gambler's ruin, which is very much like this. Let's say I flip a coin at every step, and I either gain a euro or lose a euro. Okay? So let's say I start with 100, okay? And I stop the game either when I get to 1,000 or when I get to 0. So these are my absorbing states where I stop the game. And the question is, if I start here, and again at every point I either go plus with the probability half to the right and probability left either lose one or gain one, then what is my probability of ending up here, and what's my probability of ending up here? Okay, so this is called it's a famous problem. It's called gambler's ruin. And you can solve it in exactly the same logic we had yesterday. Call the probability to get to 0 or to 1,000, it doesn't matter. Call it xi, set up a recursive equation for xi, take the continuous limit, solve the equation. Okay, it's actually easier than what we did yesterday, so it's good practice for you. Okay, going back to evolution, any questions? Okay, so going back to evolution, now we're kind of beginning to understand how it works. So if we're looking at something like trying to look at this evolutionary trajectory motivated by Lenski, then we understand that when a mutation occurs, there is this stochastic fluctuations at the beginning, and once we get to a high enough level, the deterministic equation we wrote, this logistic equation, takes over. We get essentially exponential growth. This is what we call the genetic sweep. And you see these in the Lenski experiment. So suddenly a mutant, again, think about COVID, this would be your new Omicron, sweeps and takes over the population. So usually in the pandemic, what we saw was already at this stage where it was too late. We didn't see the stochastic fluctuations. This would be some virus occurring it's somewhere and disappearing because it's not fit enough. It didn't survive these fluctuations. Okay? By the way, just 1 second. Physicists usually talk about diffusion, and drift is something like a bias. To confuse us in the context of evolution, these fluctuations are called genetic drift. So drift is what we would call a random walk in the context of evolution. Yes?
When we are analyzing the vibrations on Earth to predict the earthquakes, do we have almost the same thing going around?
No, I think it's very different. Okay, so in the same way, you can set up an equation and find the time it would take this sweep to take over the population. And to a good approximation, you can think of it as take this exponential. We saw that this is e to the lambda 1 minus lambda 2 times t, this exponential regime. And you can equate that to the population size. And that defines a time scale. And this is more or less the time scale, let's call it tau, of the sweep. Okay? So the story is we have these fluctuations. Most of the time, they go back to 0 and die out. But for each of them, with the probability that we computed here, this probability of fixing giving a fitness advantage epsilon, they take over in a time characterized by this simple relation, simply this exponential growth reaching to the population size n. Okay? So now if you look at kind of a longer time axis, what would happen is we have a genetic sweep of one mutation. Now all the population has this mutant which makes it fitter. Okay? And then at a later time, we get another mutant, right, that makes it even fitter, and it takes over the population, and another, and another. Now, the regime in which the mutation rate is small enough such that these genetic sweep are separated is called the SSWM, strong selection, weak mutation. Okay? So why? Because we see that we need mutation rate to be low enough, okay, such that these times are separated. Where do you see that you need strong selection? Just to make sure that you're understanding the jargon here. So where do you see it in the formulas? Like strong selection. Where does the strong selection enter the timescales here? So this guy here, right? This is the selective advantage of one mutation over the other. We see that if they're very close in fitness, then this increases very, very slowly. It looks something like that. So in order for them to be separated, we need weak enough mutation rate compared. There are two time scales. Mutation rates define the time scale, right? It's how many mutations do I get per unit time. And this defines a time scale. Log of n divided by lambda 1 minus lambda 2. That's the strength of the selection. Okay, so SSWM is the regime where one time scale, this time scale is much shorter than the one associated with the mutation rates. Okay? And in this time scale, which is more or less the ones we see for Lansky, mutations fix one after the other. Okay? And now we understand why, sort of what's going on here. So here we're getting mutations, and they fix, and the population increases. Okay? Why don't we see, these genetic sweeps? You will see them if you look at it at finer resolution. But this is, taking it every. You don't have the temporal resolution to resolve these sweeps. But they have been seen in the Lansky experiment. Yes?
Actually, in the other course that we have, we were talking about two oscillators oscillating at the same time. And then, we divided the angular momentum of them and summed them up to, make the equation simpler. And we had two different time scales. Can we say that here it's almost the same thing?
No, I don't think so. Time scale separation is something very generic, so it's not like, I think here we are using time scale separation in the sense that in all the calculation we did yesterday, we had one mutant, or maybe it was one mutant in the sense of one type of mutation, then it could proliferate, so it could go to 0, or it could go to 5 mutants, and so on, but it was one type of mutations. We didn't allow new mutations to arise during this regime. So we assumed already yesterday that we are in this regime of SSWM, where it's one mutation trying to fix at a time. Maybe it fails, maybe it succeeds, but we don't have others interfering. Okay? So we did rely on this. So timescale separation is something very fundamental in physics and appears in many problems, but I think the details are, you'll see it in a zillion problems as well. It's not something that's really related to the one we discussed in the other course. The alternative regime to SSWM is called clonal interference. Okay, I'll write it. We're not going to discuss it today. It's much harder to work out mathematically. And it's called clonal interference because different clones, different types, species of bacteria with different mutations interfere with each other. So you could have like something, I think we saw it during the pandemic. There was one mutant that was really going strong, and then suddenly an even better mutant came, smashed this one, and took over. Okay, so this is not something we're discussing here, this competition between different mutants at the same time. Here, we're trying it one thing at a time. Again, it's justified for a low enough mutation rate. Any other questions? Okay, so let's try to build a model to explain this. And like I said yesterday, it's going to be a null model that's actually going to fail and help us reveal what sort of model we do need to understand this slow increase in the fitness in the Lenski experiment. So one lesson is, let's say I'm setting up a model. My goal in solving the model is not necessarily to try to explain the data. It's a successful model as well if it fails. It's OK to have this is my null model, my simplest model to explain this. Let's see if it works. Okay, so the goal of models is not to capture every detail of reality necessarily. It's to kind of tell us what's going on, what could be working, what might be missing. Okay, so this will be clearer as we go. So let me kind of ask you, if we want to have a model to explain how the fitness increases quantitatively in this evolution experiment, what can anybody give me a model? Doesn't have to be right. Just define any model. Any ideas? Model, it's always like at this stage a bit vague. So it's like, it's not a mathematical problem that I ask you to solve, right? It's like, I want to understand. We see that in the Lenski experiment, fitness increases as a power law with a power very close to 0. So like time to the power of minus 0.1, essentially a logarithmic increase. How can I capture something like this? How can I try to capture it? Okay, so let's think of the genome. Okay, so I have like my long, my double helix, and I have a long sequence. And for simplicity, let's think about a binary genome. You can make it a 4-letter code. It won't matter. Okay, so let's say I have like 0, 1, 1, 0, and so on. And let's say that at every point, there is the right sequence that would contribute more to the, maybe I want to have a 0 at this point in the genome, but I want to have a 1 at this point in the genome. Okay? So it's like a spin system where there is a preferred orientation at every site for the spin to be either up or down. Okay? So this is kind of a minimal model if you want. Okay? So then if I start with some genome, my question to you is it a hard problem to solve? Is it an easy problem to solve? What do I need to do, really, in order to get to kind of the maximal fitness? Yes? Very good. So we'll get to that. So let's assume, as our null model, that there are no interactions between the sites and see how far we can get to. Okay? Once we put interactions, it's going to be super complicated. Okay? So no interactions for now. Yes?
Sorry, can you just repeat what are the zeros and one?
Genetic code. Okay, so this is A, CTG, but now, I don't really care about that, so I'm just saying let's replace it with zeros and ones. And let's say that the fitness is a sum of some coefficients AI times SI, where SI is AI will be the contribution of a spin. The spin can be, say, 0 or 1, or 1 and minus 1, it doesn't matter. And AI will be some coefficients. Okay, how much this contributes to the fitness and whether this is positive or negative will tell me if I want the spin to be in the 1 state or in the 0 state.
So in this picture, like the reality of the DNA would be a Potts model, and we're considering a new model.
Yes, and it doesn't matter. It would be very equally easy to do the calculation for the Potts model. Okay, yes?
So we're trying to reach a specific genome?
So in this case, let's say I tell you that which genome is the best genome, if this is my fitness. This is easy to figure out, right? The only thing you can do is choose the sign correctly. So you want the S's to be of the same sign of the AIs. If the AIs are positive, you want the SIs to be positive. Let's say it's plus minus 1. I think it'll be easier. And if the AIs are negative, you want the SIs to be negative as well. So you get a positive contribution to the fitness. But the key point is, in this null model, optimizing fitness is very easy. Why is it very easy? It's one hill. It's one hill we're climbing. Okay, there are no metastable states. We're just climbing one big hill, and we're optimizing the spins one by one. Okay, so what will happen here? I get I start with some genome, and maybe this is already in good place. This is already in good place. This is already in good place. Maybe all of these are fine, but say this one is supposed to be a -1. I want it to be a -1. Okay. So what will happen? I will get a mutation here. I get a -1 here. And then what will determine whether this will fix or not? Exactly. So what determines the energy? If this is site 17, it's the coefficient A17. If it's large, then it contributes a lot to the fitness, and I will get, there is a good chance of fixing. And if it's very small, then even if I get this mutation, I will have to get it many, many, many times until I finally fix it. This is what we learned yesterday about fixation. So you can see that now in this model, the distributions of A's, if I look at these A coefficients, the distribution of P of A, let's say it's some Gaussian, that will matter a lot. Okay? And which ones will I fix first, and which ones will remain for last? More or less? Exactly. So like really what we care about is the magnitude of A. So maybe I should plot the histogram of the magnitude of A. First I fix all these guys. And then the ones that will really contribute to the long time behavior are the behavior near for the small a's. Okay? So now like we've made progress. We defined a model. We understand that even this entire distribution, we don't really care about it at long times. At long times, we care only about the behavior here for the small a's. So we can take this to be maybe a constant, assuming that there is nothing special about this distribution, right? Even if it's an exponential and so on, as long as there is a finite value at here, I can just take this to be some constant. And now for this model, we can calculate what would be the fitness as a function of time. Is this clear? Okay, so again, it's the wrong model, we'll see, because it won't capture this very slow increase in the fitness. This will give us a much rapid increase in the fitness as a function of time. Why is it faster here? It's faster because, again, we're doing this convex problem that we're just climbing one hill and there are no metastable states, and we'll get to that interactions part later on. But it's still useful to see, like, maybe this null model would have been good in some case where the mutation rate is slow enough or things like that. But we'll see it's not the case. Yes?
I don't really get how it is, like, if this is statistical mechanics inspired or not. Like, if a mutation raises energy, I need some kind of part of the entropy of temperature to understand if we will go to fixation or not.
so this is not energy. Okay, we're soon going to kind of use this analogy with an energy landscape more systematically. But for now, fitness has nothing to do with energy or entropy in the statistical mechanics sense. Fitness is how fast the cells grow, for instance. We spoke about yesterday about this E. coli growing, being diluted, growing, being diluted. Okay? So, you know, these E. coli are hugely out of equilibrium. There is no way you can apply statistical mechanics to them per se. Don't, you know, I'm using an energy landscape in the loose sense, not in the real sense. Okay? But soon it's going to be even more useful, this analogy with thinking about a glassy landscape and things like that. So bear with me. You'll see it in a few slides. But again, it's not, there is no temperature. Temperature is not, I mean, of course, for the bacteria, temperature is relevant. But it's not like thermal fluctuations or something like that. It's not, that won't work. Bacteria are too far out of equilibrium. Okay, they're spending you know, ATPs like crazy all the time. So let's say I got a mutation at this site. Okay, so now if this site was 1 and I made it a -1, there are two options. If the A700 corresponding to this site was already positive, then what do we call these kind of mutations? Deleterious. They're bad for the population. What is the chance of it fixing, assuming we have a large population like we do in the Lenski experiment, 10 to the 6? 0. Okay, so then we got a mutation and it gets kicked out, done. Okay, and if it was positive, then now we're kind of using all the machinery we did yesterday. It's already, we're using it. We know that the result is that it fixes with a probability that is related to this coefficient A700. So if this mutation, if this gave me a change in the fitness of 2%, I have a 2% chance of fixing. So maybe if I run a simulation 50 times, it'll take me on average until I fix this one. Okay, but it's not so bad, because eventually it'll happen. And we do get a lot of mutations all the time. Okay? So this sort of problem is a bit like a famous problem called the coupon collector problem. Okay? So the coupon collector problem is I have a deck of n cards. Okay, it's not a deck. There are n different types of cards, and I'm collecting them, and every time I get a random card. And the question is, how long until I have all the cards in the collection? I'm sure you collected cards as kids or something like that. Okay? So again, it's not a difficult problem because you cannot, once you get a card, you have it. You don't lose it. Same here. Okay? So in this case, also the answer is it's not so bad. Like if you get super lucky, it's going to take n. But you might think a priori that it'll take you much, much longer until you have all the different cards. But the answer is it's not so bad, and it's kind of a two-line calculation, right? So the first card you get is unique. You never had any cards like that. So to get the first card, it takes you 1 round. Well, in the next round, there are n-1 new cards you can get out of n. So what's the probability of getting a new card in the second round? n-1 over n. So how long, on average, until I get the second card? Well, the probability that it's new, it's n-1 over n. So how on average, what's the expectation value until I get the second card, which is new? Excellent. Why? Because just like when you throw a die, the probability of getting 6 is 1 over 6. How long do you have to throw it until you get 6? 1 over 6, which is 6. Okay, same story. It's 1 over the probability. That's the expectation value. So by the same rationale, what's to get the third new card? How long do I have to wait on average? n over n-2, and so on and so forth, until for the last one, okay, now it's just the probability is 1 over n to get the new card. So it's n, right? So now you can sum up this series very easily. You can convert 1 over x to an integral, and you will see that this essentially is approximately equal to n log n. So you can see it's an easy problem. Okay, it takes longer than n, but it's not even n squared. It's just n log n. It's not too bad. So we have something similar here. We're going to see that collecting now the mutations we need. They're all independent of each other. We just need to collect all the coupons. It's not so bad. What makes this problem a bit different than this problem is the fact that we have this extra ingredient of fixation probability. When we get a mutation, if it has a small fitness effect, we don't necessarily keep the card. We have to try many, many times until we get to keep the card. This is this fixation probability. Okay? So now we'll do an easy calculation, putting things together, taking this simple model, taking the fixation probability, and finding fitness as a function of time for this null model that turns out to be wrong. Yes? It'll be clear. I won't assume it in the beginning of the calculation, and I'll show you in the calculation where it enters. But the intuition is these are the hardest cards to get. These cards, I get uniformly all of these mutations everywhere. But these I get to keep because they have a big effect, so they fix. This one, if it has a tiny effect, I need to get many times the mutation until I finally fix it. So these are the ones that at long times will play the major role. Okay? But again, you'll see it in the calculation. And this is also, you'll find it in the notes that I uploaded. Keep in mind, in the notes that I uploaded, there is stuff that I don't talk about here. So you don't need to read it unless you're interested, like fixation probability in the case of a serial dilution experiment. And it's a harder calculation than the one we did in class. And there is also something very interesting about something called Hardy-Weinberg equilibrium that I'm not going to discuss today because it's not about, it's interesting, but it's not so much about bacteria, so I decided not to speak about it in this course. Okay, so we understand that we have some distribution of fitness effects, and I can look at the sites which need to be optimized. All the sites that are already optimal, I don't care about because they're already in place. And I can say maybe I have n of S and T is going to be the number of mutation, the distribution of the sites with mutation effects S at time T. Okay? But actually, I don't want the probability distribution. I want the total number. Okay? So the fitness will be some constant minus an integral from 0 to infinity, n of S and T times S ds. Okay, let's understand this. So these are all the mutations of fitness effect S that still needs to be optimized. At time t equals 0, I need to optimize all of them. As time goes by, I'm going to decrease their number because I fixed things. And the fitness is going to increase by an amount S for each of them that I fixed. Is this clear? Okay, so let's say I have at the beginning a mutation with effect S1 that needs to be fixed, and another one with S2, S3, S4, and so on. I make a histogram of them. Okay, so now S is my axis, and n of S and T is how many I have with a fitness effect within some region. Okay, now I take the continuous limit, so I have some distribution telling me if I take n of S and T times delta S, it tells me in this regime between S and S plus delta S, this is the number of mutations that need to be fixed. Okay, so if I fixed some of them, what is the effect on the fitness? The lower I lower this, the better the fitness is. For each one of them, it's n times S is the effect on the fitness, because S is the magnitude of the fitness effect. Okay? Now, if I look at df to dt, it's simply equal to minus this integral of dn to dt S ds, right? This is the rate at which I'm fixing mutations of a given magnitude. Okay? But we know what dn to dt is. This is what we did yesterday. So if I look at dn of S and T to dt. What is that going to be? It's going to be negative because I only fix mutations, so I lower the ones that are needed to be fixed. What is the proportionality constant here? Let's try to work this out together, okay? So these are the mutations that need to be fixed of a given magnitude S. What is the rate at which I'm fixing them? First, I need to have a mutation rate. So this is my mutation rate. This is the rate at which mutations are happening. Then what else is it going to be proportional to? S, because of the fixation probability. So this is the P fixation as a function of S. And lastly, what factor am I missing? No, because it's every mutation works for itself. It's independent of the others. Not neutral. The fact that we're not neutral enters here, that it's proportional to the fitness. Large population, not neutral. There is a fitness effect S. S is the selection advantage. But there is a kind of trivial effect I'm missing? N, right? So it must be times N, because the more sites I have to fix, the larger the rate. So this is the equation for fixing mutations, and that's going to enter into here. And now, finally, we have an equation for the fitness. So we find that the time derivative of fitness is equal to minus mu times an integral from 0 to infinity. Okay? And now, this we can solve. Right? So the solution of this is simply an exponential. So every mutation of a given effect obeys n of s and t equals to n of s and 0, e to the minus mu times s times t. What does that tell us? This tells us something very simple. They all decay exponentially with a time scale that is inversely related to the mutation rate, but also to their fitness effect. This is what we said. The smaller the fitness effect, the longer it's going to take me to fix these. So now I can take this, take the derivative, plug it into here, and I will get minus mu n of s and 0, e to the minus mu t mu st times s squared, because I take the derivative, times ds. So we're almost done. And now, is this clear? Any questions about this calculation? Yes?
I lost what's S.
S is the fitness effect. So we have, initially, we need to know what are the mutations that need to be fixed. So S is the fitness effect of a given mutation, and we start with some distribution. N of S at time t equals 0 is what needs to be fixed in the first place. These are all the mutations that I need to fix for a given fitness effect.
So the notation is different with the left blackboard,
It's almost the same story. It's almost the same, right? Because S is essentially proportional to A. The distribution of the A's are the distribution of the S. It's the same thing, right? If I know the distribution of the coefficient A's, I know the distribution of mutations that need to be fixed. So it's almost the same thing.
So S is basically the sum of fitness effect of what needs to be.
Yes, that's the only difference between them. That A, I don't know which ones are already in the right place and which ones need to be fixed. So the distribution of A's may look like that, but maybe this, and this are already fixed, so I don't need to touch them. So N of S are the A's that need to be fixed. Okay, so it's a kind of very minor detail. Okay, so now we see that at long times, and this is the question that was asked, what happens at long times? At long times, all the exponentials decay fast, except for the ones corresponding to small s. Okay, and now I can make an approximation where I take this, say, Gaussian or whatever distribution it is, and I approximate it by this constant value at small values of A. So I can approximate this by n of 0 at time equals 0. So this is just a constant. And then I find that df to dt is approximately equal to minus the mutation rate times an integral of e to the minus mu st s squared ds. This is an easy integral you can all do. And you can see it from dimensional analysis. Okay, the dimensions here, s has dimensions of 1 over time. Okay, or so this must scale as 1 over t cubed. You can just do the integral. So if the derivative of fitness goes as 1 over t cubed, we find finally that fitness is going to be some constant a minus another constant over t squared. So we expect within this null model that as time goes by, fitness will increase to a plateau, but this dependence is 1 over t squared. And it's too fast compared to what the experiment shows. Yes?
But we don't want that.
We don't want that, but what does it tell us? It tells us this model doesn't work. So what's going on? What's going on? First of all, it's an open question. Okay, so people don't understand the Lenski experiment that well. Okay, but we can hypothesize. What do you think is a natural place to assume that we've oversimplified the problem and kind of, and you already said it. It's interactions between genes, right? So this model assumed that we're collecting coupons, right? That every gene is independent of every other gene, and we're kind of optimizing them one by one. And it gave us 1 over t squared. You know, it's a power law, so it's not like super fast, but it's not slow enough compared to the experiment. And the natural way to kind of go beyond to the next step of the model is to say, maybe we can account, just 1 second, for interactions between genes. What do I mean by interactions between genes? It's known in biology that if I make one mutation here and another mutation here, the total effect on fitness doesn't have to be the sum of the two mutations. It can be less than the sum. It can be more than the sum. There are interactions. So you can say that the fitness as a function of the state S1 and S2 is not simply A1 S1 plus A2 S2, which would be the case in this simple model we analyzed, but it can also have an effect, let's call it A12 or coefficient B12 times S1 and S2. These are interactions between genes. In the context of evolution, interaction between genes is called epistasis. So you can measure it. You can take bacteria or yeast, make a mutation here, make another mutation here, make a double. So you measure the growth rate when you have just this mutation, or both mutations, and see whether these interactions exist or not. And you find that sometimes they're positive, synergistic, and sometimes they're negative. There was a question back there. Very good. So you probably could, but then you would have to. So first of all, it's an excellent comment. So you can say, maybe my model is different, and I need to have n. So how would I get something slower? I will need to have n vanish at very small fitnesses. Then the question I would ask you is, like, why would you have a distribution of fitness effect that penalizes very small fitness effect? It seems unlikely, unless you come up with an explanation, right? So it's what I would call a cooked-up solution, not a natural one. So we can discuss it more, but there is no reason a priori that the distribution of, no experimental evidence that the distribution of fitness effect would kind of be something that vanishes at small fitnesses. And it would have to vanish very fast, right? It's like having a gap. It's like we're not allowing small fitness effect mutate. Why? In fact, the opposite is probably true. Most mutations are near neutral, and it's very rare to have mutations with a big fitness effect. So something like a Gaussian or exponential is more plausible. It's a whole field. There are papers on what is the distribution of beneficial mutations. We can discuss it over the break further. Other questions, comments? Yes?
Behind you, it is B or
here. Oh, this is just, I'm using here the result that the fixation probability as a function of S was equal to S. This is what we proved. This is what we worked hard yesterday to get. Okay? So this is what we conclude. We concluded that slow relaxation hint that epistasis is important in understanding this fitness as a function of time. And that suggests the next order model. And again, this is an open problem. I'm not going to kind of discuss it in much detail today. But if you take a more complex model where fitness is the sum of the point effect of individual mutations, this is this AI-SI we had now. But now we add random interactions between genes. Okay, so the simplest model would be, let's assume that genes interact randomly with each other. It doesn't have to be that way. It's, again, a simplifying model. Reminiscence of maybe stuff you'll see in ecology by Amir next week. He's going to discuss random interactions between species as a null model for ecology that's going to give some interesting results. So in the same way, this is what people would call a spin glass model, and this is where the analogy to statistical physics is even more helpful. Okay, so this type of model has been written for the first time to understand magnetic properties of materials where spins interact with each other, not really randomly, but in a distance-dependent way that changes sign. So sometimes it's positive, sometimes it's negative. And so a very famous model says, OK, let's just study the behavior where the interactions are Gaussian and drawn from a Gaussian, so they can be positive or negative, and chosen randomly and independent of each other. This is something called the Sherrington-Kirkpatrick model. It looks very simple. It took a long time for people to solve. And one of the major advances was by Giorgio Parisi that got a Nobel Prize a couple of years ago, among other things, for developing these mathematical techniques to deal with understanding these spin glasses analytically. So they're much, much harder than the stuff we're doing this week. And so you can say, what if we add random interactions between genes? How would that affect the fitness trajectories? I'm not going to discuss it here today. We have 2 papers on this. And we show that this can indeed give you a much slower increase of the fitness. In some cases, it'll give you a power law increase, but with a power much smaller than 2. In another limit, it can give you logarithmic dependencies. I'm not claiming that this is necessarily the explanation for what's happening in the Lansky experiment, but we do think that there is a lot of evidence that epistasis must be taken into account in order to understand these features of the Lansky experiment. And we're actually working now on a paper to discuss something else related to the Lansky experiment that was done, motivated by experiments done about a year or 2 ago. So in these experiments, they took mutations that were beneficial early on in the experiment. So take this graph on the right, take cells, take bacteria from that generation, and measure the effects of it wasn't exactly single point mutations, it was what happens when you delete a gene. So you take many genes, you delete them, and you measure the effect on fitness, and you get some genes are beneficial, most genes are deleterious, right? Like most genes, if you delete them, it will be bad for the bacteria. But some, you delete them, and it's better for the bacteria. Why? Because you give them a particular medium, and maybe they don't need this gene in that particular medium, so they can live without it, then it's better to get rid of it. Okay? Now they took cells from later in the evolution, and they did the same thing. They deleted the same genes and measured the effect. And what they found is that gene deletions that were beneficial in the past can become deleterious in the future, and gene deletions that were deleterious in the past can become beneficial in the future. Okay, so the effects of genes being beneficial or deleterious changes throughout the course of evolution. And again, this won't be captured by this model. In this model, this doesn't change. It's just some number that if you're beneficial, you're always going to be beneficial. If you want this to be up, you always want it to be up. But if you have these random interactions, Bij, SISJ, then as evolution progresses and you're flipping some of the spins, suddenly something that was beneficial can become deleterious, and something that was deleterious can become beneficial because of these genetic interactions between the genes, because of epistasis. So we show that that can capture, this simple model can capture the statistics of this effect that we call scrambling in the Lenski experiment. Okay? And now I'm going to kind of almost switch gears, and I'll show you one last thing about the Lenski experiment, and that will be a transition to the second topic in the course. I wrote that initially that we have 3 topics, but I think it's probably we're going to just have time for 2 topics. So the first topic was evolution, and I hope you got a flavor of the sort of math and physics that people do in evolution. And the second topic is going to be about cell size control. One last thing I forgot to mention, and this was related to a comment you had back there about the statistical physics. When you think about adding these interactions, the landscape becomes glassy. And this is where this analogy with statistical physics becomes helpful. When I don't have this term, we know that the problem is climbing one big hill. I have to optimize and just collect the coupons. When I have this, I get many, many metastable states. These are states where flipping one of these spins is not enough. I flip the spins, and I'm done. I got to a local maximum. Now, in order to become better, what do I need to do? Flip 2 spins or 3 spins at the same time, and that's much harder. Okay, so there also works on something called stochastic tunneling, where you have to we can calculate what is the chance of getting from this state to this state where I need to kind of flip 2 spins. I need to get a deleterious mutation, and before it dies out, I need to get another mutation such that only the combination of both mutations will be beneficial. Okay? So this is this intuition of a glassy landscape is sometimes helpful when you think about these things. Okay, so the last thing I wanted to tell you about the Lenski experiment, yes? These are all time derivatives. What's that? This one? So the question, if this is a time derivative? Yes. So we solved the n, and we found that n decays exponentially. We plugged this exponential dependence into the formula for the time derivative. But the integral here is over s, so it's fine to put the kind of time dependent. We can discuss it offline. Okay? All right. So the last thing I want to mention about the Lenski experiment is this curious fact already observed 6 years into the experiment. The experiment started in 1988, and 6 years into the experiment, they published this result that fitness increases over time. They don't have the resolution they had it. Remember, in the previous plot, it was 2013. They had 50,000 generations or 70,000. I don't remember, so they had a lot more. Here, it's only 10,000 generations, so they don't have such a good quality, but they see the fitness increasing in time, and they see cells are becoming bigger as a function of time. Okay? So the sort of questions you can ask is maybe cells are selected because they're bigger. Maybe the mutation made the cells bigger, and that's why they're fitter. Is bigger fitter? Or are cells growing faster, and for some other reason that makes them larger because of cell properties? Okay? So we're going to kind of discuss this at the end, maybe at the last lecture. We'll maybe come back to it. And I want to now transition and talk about these questions of how cells control their size, what determines cell size, and maybe how we can explain this fact that cells that are growing faster are also going to be bigger, and try to understand the causal relations. Like, is the arrow going from left to right, or is the arrow going from right to left? Okay? And by the way, maybe I'll mention there is a concept in evolution that's worth knowing about, and it's called the spandrels. Okay? Has anybody heard of this? Okay. So spandrel is if you, this is an influential paper from a long time ago called The Spandrels of San Marco, a critic of the adaptationist program. So San Marco's cathedral is about 2 hours away from here in Venice. And the story that they outline here is that you come to this cathedral and you see beautiful decorations. The most beautiful decorations are here. This is called a spandrel. Okay? So the spandrel it's an architectural element, which is the part between the dome and the arcs. Okay? And then you come and, we as scientists come and would say a story like, oh, look at these beautiful drawings. Maybe they build the entire building to accommodate for these beautiful drawings on the spandrels. And then when you think about it, you understand that it's exactly the opposite, right? That you put a dome on arcs, and you're left with these spaces. You have to have these spaces, right? And then you do something with them. Okay, so the causality is the opposite. So a lot of times in evolutionary biology, you would see something and you say, oh, this must have been adapted to do this and this and this. Well, it's something completely different. You evolved something, right? Evolution works in a kind of funky, nonlinear way. And then you're stuck with some artifact and you make do with it. So you do something with it. But it's just because you kind of came to that. Okay? So in the same way, I will suggest that it's not that cells are selecting, it's not that size is important and cells are fitter because they're bigger, but it could be that cells are bigger as an artifact of something that's going on that has to do with their internal way that they will regulate size. So this will be clearer after we discuss cell size control. Okay, but I think in general, it's important to know this concept of a spandrel in evolution. That things are, because the way evolution works, things are much more complex. And when you see something, your intuition of putting an adaptation hat on top of it, this has adapted to do this and this and this, might be wrong because you need to know the whole historical contingency of how you got to that point in space and time. Okay, so that's that for evolution. And now we're going to talk about cell size. And let me switch presentation. Okay, and it's also going to be about learning from fluctuations in the sense of the Luria-Delbruck experiments that we started out with. So here is another video of E. coli, just to remind you, this is how E. coli looks like. It's growing and dividing in a microfluidic chamber that keeps it at a constant condition. So it's a different setup than the Lansky experiment. Here you're constantly feeding the cells, so they're growing and dividing, and you can track them for hundreds of generations if you wish. And like we said, the cells double in length. And after they double in length, we discuss this MREB that helps them divide very symmetrically. After they double in length, this protein forms filaments. It's called FTSZ. And then this ring at the mid cell shrinks and shrinks and shrinks until one cell becomes two daughter cells. And again, if you do it in good growth conditions, then this can take as short a time as 20 minutes. Okay, so it's quite amazing. You double yourself in 20 minutes. And you do so, by the way, under an enormous, I won't talk about it in this course, but there is interesting physics to do with the mechanics of these bacteria. The pressure inside them, the turgor pressure, due to there is a larger concentration of molecules inside the cell than outside. And that creates a pressure called the turgor pressure. And in E. coli, it's of the order of 1 atmosphere, which is enormous. In other bacteria, like B. subtilis, it's a soil bacteria. It has a thicker cell wall. The turgor pressure is 20 atmosphere. Okay, that's about the pressure of a road bike. Okay, so they're really highly pressurized. And by the way, that makes them vulnerable. So some of the antibiotics, what they do, actually a lot of antibiotics, all the penicillin family that I'm sure you've taken at some point in your life, what they do is they damage the cell wall production. The cell wall is this capsule that makes the cells rigid and fights against this turgor pressure. If you damage cell wall, the cell blows up. Okay? It's a process called lysis. Okay? So and also that's what some viruses do. mess up with cell wall production and the cells blows up. Okay? So in all of this growth, you have hundreds of different proteins interacting with each other in a very complicated way to create a very organized behavior. And one way that we can quantify this organized behavior is by looking at the distributions of the cell size. So if I take this video, literally this video, and I quantify the points at which cells are born and which cells are divided, and I make a histogram of the distribution of cell size at birth and division, I get very narrow distributions both for birth and for division. Okay, so I can quantify distributions, for instance, by their coefficient of variation. Can quantify random variables by the coefficient of variation. That's the standard deviation divided by the mean. So it's a dimensionless number that tells me how tight the distribution is. And for cell size in E. coli in fast growth, you get numbers of the order of 10%. So it's very narrow, right? It's not something, it's something you need to work hard for to get such a tight distribution. And we're going to quantify today, what do I mean by, or what do I mean by tight? And for instance, we'll see that in a model where cells simply time their divisions, let's say I divide every 20 minutes, we will never get something like this. We'll get something infinitely broad, and we won't be able to get something so tight. And you'll see what I mean in a couple of minutes. Any questions? Okay. I'll tell you one more thing about bacteria. I won't go deeply into it this week, but it's kind of a mind-boggling phenomena that's super interesting. And this is the following fact. If you grow bacteria in slow conditions, we've already mentioned the circular chromosomes with the origin and terminus that you duplicate, right? So in slow growth conditions, I take my chromosome. It's this circle. I start at the origin, I finish at the terminus, and I have two replication forks, one going to the clockwise and the other going counterclockwise. And when they meet, then I basically duplicated my entire chromosomes. I have two circles. I can split them, and I have two intact chromosomes. So the cell grows, duplicates its chromosome, and divides. Perfect. Okay? Maybe this is how you'd imagine it. What happens in fast growth is quite mind-boggling. In fast growth conditions, the time it takes to duplicate the chromosome can be longer than the time it takes the cell to divide. So the cells divide every 20 minutes, but the time it takes them to duplicate the DNA in these conditions is 40 minutes. And now you scratch your head and you say, how can that be possible, right? So this can be possible if you start duplicating DNA already for your grandchildren or great-grandchildren. So when you're born, you're already inheriting partly replicated DNA that was started by your parents, by your parent cells. And you're going to start duplicating DNA for your grandchildren or great-grandchildren. So these are called multiple replication forks. So if you look at the DNA in these conditions, it's not going to look like simply this two bunny ears on top of the chromosomes like I drew on the board. You're going to have bunny ears on top of bunny ears on top of bunny ears, these nested replication forks, which is sort of crazy. Okay? And that leads to additional interesting questions. How do cells know when to start replicating a new round of DNA to make sure that every cell doesn't have too much DNA and doesn't have too little DNA? Okay? Now, the first thing I want to do in kind of building, and I think tomorrow we'll do most of this, but I want to show you that a null model where cells grow for a fixed time doesn't work in controlling cell size. So again, if I'm giving my E. coli cells a good clock, and I tell them try to divide every 20 minutes, this will be a problem. Okay? We're going to see why in a second. And the problem relies on the fact that cells grow exponentially, approximately exponentially at the single cell level. So a population of cells for sure grows exponentially. That's not surprising. But also if you look at an E. coli cell, if you look at its length or volume or biomass as a function of time, it increases exponentially at the single cell level. Again, approximately. Can anybody suggest why that might be the case? And that's related to previous courses you took in this spring college. What feature of a cell makes exponential growth a natural thing to expect for E. coli? Concentration of what? Proteins, okay, and which proteins in particular? I mean, what's that? Which is the kind of the protein that I would like to talk about when I think about exponential growth of E. coli? Ribosomes. And what feature of the ribosomes would make exponential a natural expectation? Good. So what makes ribosomes? Ribosomes. Okay, so they're autocatalytic. Ribosomes make ribosome, right? And then if I look at the number of ribosomes in an E. coli cell, I will write the simplest equation would be, again, coarse graining transcription, which is a long story, but I can write dr to dt is proportional to r. The solution of that is exponential and leads to this growth law that the fraction that we discussed, I'm sure you discussed with Jacopo, right, that the fraction of ribosomes in the proton is proportional to the growth rate. So that comes from the simple equation saying ribosomes are autocatalytic. They make ribosomes. Dr to dt is proportional to r. Okay? So we expect exponential growth for E. coli, and there is evidence that this is approximately the case. We have papers talking about deviations from that. It's a long story. I won't get into it. Okay? But to a first approximation, it's true. And if that is the case, we're going to see we have a problem. Okay? So let's see what the problem is. The problem is if we look at this lineage tree, so if you look at this family tree, I start with one E. coli, it becomes 2 cells and 4 cells and so on and so forth. If we follow one lineage, so I'm following the blue line here on the right, and I look at the cell size at birth. Okay? So I look at the cell size at birth at the first generation, second generation, third generation, and so on and so forth. Then I get an equation that Vn plus 1, and every factor here has a lot of biology in it. So we'll discuss it in a second. Vn plus 1 is equal to Vn times e to the lambda t. This just encapsulates what we just discussed, that cells grow exponentially at the single cell level. The whole ribosome picture is in this simple formula here. So you understand that, right? And then there is also a half, which is also something we discussed. Why can I write a half here? Split symmetrically, the min system, again, a ton of biology making it divide symmetrically. It's not something that's obvious. Yes? V is the volume of the cell at birth. I want to look at the volume just when it was born. Okay? So we get this equation. Vn plus 1 equals half Vn e to the lambda t. Okay? Now, lambda, again, is a long story. It's the growth rate. Let's neglect fluctuations in the growth rate. Let's for now assume that cells are growing perfectly exponential with no fluctuations. Okay? So a priori, if you haven't taken in physics courses, you might think this is fine. All you need to do is make sure that you grow for a time that makes e to the lambda t exactly equal to 2. Okay, so if I grow at some growth rate, I will grow for a time that is log 2 divided by the growth rate. Perfect. E to the lambda t is 2. Every time I double, divide, double, divide, double, divide. What's wrong with this picture? I want you to think about it because this is key. Yes, t is the time between birth and division. And I'm asking what's wrong with this picture where I simply, have a growth rate which is, I double, mass doubling time of 20 minutes. So I make sure that I divide every 20 minutes. I have a clock. I time my divisions to be every 20 minutes. Seems fine. What's wrong with it? But in a perfect world, this would work, right? I'm born at a volume of 1 micron cubed. I double, divide, double, divide. Why does this timer-based picture, why is it going to, we know it's going to fail. What could be the problem? We don't know that because we haven't calculated, but there is a more fundamental problem. Stochasticity. What would stochasticity do?
The time for dividing and doubling will not be exactly the same.
Okay. So let's say it's like some narrow Gaussian around 20 minutes. So let's say it's 20 minutes plus minus 1. Well, sometimes it's more, sometimes it's less. What's the problem? Okay, I'm glad that it's not obvious to you. Let's kind of look into it. Okay? So let's take this equation, and let's take the logarithm of both sides. Okay? So we have Vn plus 1 equals half Vn e to the lambda t. And I want to work with the variable xn, which is the logarithm of Vn. If you want, you can divide by the typical size. It doesn't matter if you don't like a logarithm of something with dimensions. Okay? So we'll find that xn plus 1 is equal to xn plus lambda t minus log of 2. Now, I can choose t. Let's say t is a Gaussian. And I choose the average. It's a shifted Gaussian. It has a mean of 20 minutes and some standard deviation around it. So I can choose the, t such that this, on average, has a mean of 0. Okay, I choose the time on average to be exactly such that I double. Fine. But now this is going to have some finite variance, right? Because maybe the variance is 2 seconds, but there has to be some imperfections in my timing. So there is some finite variance. So if this is a Gaussian with some finite variance and mean 0, what is this equation that we obtained here? Random walk. So what's going to happen over many, many generations? What kind of distribution are we getting? Gaussian. And what is the variance? It increases with time. Every generation, it builds up and builds up and builds up. So over time, cells are still like positive volume because this is a logarithm, but the logarithm of size will become broader and broader and broader. So I will get, after some generations, huge cells and very small cells. Fluctuations within this model will build up over time. So again, it's all about the stochasticity and the fluctuations, and we can see that this is a terrible model that is not going to work. It's not that it doesn't fit the distribution. It doesn't lead to any distribution. It doesn't lead to a stationary distribution. It leads to a time-dependent distribution that becomes broader and broader and broader over time. Okay, so this is the problem. And let's talk a little bit about diffusion and random walks. Again, I could spend the whole week talking about the exotic properties of random walks, and it's related to the first passage time problems we discussed in the context of evolution. Very interesting mathematics. We're just going to kind of give you a little bit of flavor of that. And then we're going to fix it. So random walks, you can study them. This is an example of a 1D random walk, but you can study them in any dimensions, right? And this is, as far as I can tell, the first proper formulation of this problem, of literally the problem of the random walk. And this was done by Pearson, who was an important statistician in the 20th century. And he formulated this problem where he looked in 2 dimensions. And he basically said, who here has heard of random walks? OK, so I'll go very quickly. So you know, he formulated the problem where you basically take a number of steps, change the angle to some random angle, and take another fixed number of steps. And you do this many, many, many times. And you ask, what is the distribution after a large number of steps? It's not a very difficult problem to solve. For some reason, he couldn't solve it. This was a letter he sent to a Nature magazine asking for help in solving this problem. Actually, the person that answered and solved it was Rayleigh, Lord Rayleigh, who also made a lot of important contributions to physics. But kind of surprisingly, in the same year, I think this is 1904, Einstein developed independently the whole formulations of random walks. Okay, you see it at the back of the room, World Year of Physics 2005. I'm sure they talk about random walks there. And so Einstein developed a general formulation of random walks, proving some of their key properties. Okay, so this was Rayleigh that solved it. I'll get to this in a second. So Einstein proved that the length scale associated with diffusion goes as square root of time. This is an important property. And he showed that the distribution is Gaussian. So you basically get a Gaussian, the probability distribution is going to be, say for our case, at long times, what we'll get is that the logarithm of size will follow a Gaussian with a width that scales as square root of time. So if I took 1,000 generations, I will get a very broad Gaussian, clearly not in agreement with what we see experimentally in these tight distributions. Okay, so these are important properties of diffusion. I'm going to diverge for a second for cell size, because once we're talking about this, I think it's important to tell that a few years after Einstein's predictions from 1905, Perrin made a beautiful experiment where he basically, one of the parts of these experiments was to take small spherical beads, okay, I think made of mastic, of gum, and suspending them in a liquid and looking at the random Brownian motion of these particles. Okay? And Einstein gave us the tools to understand that we expect that this is the data. This is actually the data from his experiment. And you see these random motions. These are literally the random walks of these small particles. And from analysis of basically this nonsense, again, it's like Luria-Delbruck. You're learning from the fluctuations. If you just look at it, you say, OK, it's some random fluctuations. What can I do from it? But actually, based on the theory, we know that what we need to do is to look at the mean square displacement. And we'll find that it's proportional to time. The squared of the displacement should go as diffusion constant times time. This is what he found. And if you look at the distribution, he found that it's approximately Gaussian, just as the theory predicted. So what did Perrin gain from this? He could prove for the first time that matter is made of atoms. This was the proof. It's kind of crazy. It's only the beginning of the 20th century. And this is the first time that we proved that matter is made of atoms. And we could calculate, Perrin could calculate, based on this data, Avogadro's number, which tells you how many molecules you have in X grams of oxygen or whatever material you have. So you prove that matter is made of atoms, and you counted how many atoms you have or molecules for a given amount of material. And for that, he got a Nobel Prize. Okay, so it's a very important result that built on Einstein's very simple theory of random walks. And in fact, if you're interested in the details, so I can give you two references. One is this popular science book. I already recommended one in the beginning of the week on bacteria. This is another popular science book all about random walks and the physics of random walks and why they're important for things from behavior of the stock market. And there's something called the Black-Scholes model that also builds on random walks to the atomistic nature of matter, and as well as many other things. So this is a popular science book on random walks. And if you're interested in more technical details, this is my book that I mentioned perhaps earlier in the week. And I basically follow Einstein's original derivation. It's very simple. Einstein's original derivation of the diffusion equation, starting from random walk in any dimension and ending with a diffusion equation, from which it's easy to prove this Gaussian and this square root of t scaling that is important. Okay, so if you want to see the details, you can find it there. Okay, so going back to our size control, we understand that this timer-based model is not going to work. And let's kind of, just for fun, go through some simulation to see the problem and to see how we can fix it. Okay, so I hope you can see this, this code. It's very simple. So in the first part, what we're going to do is simply simulate this random walk that we wrote on the board. So we have some variable xi's. This is, if you want, the logarithm of cell size. And in the next generation, I'm simply adding to it 0.05 times a Gaussian random variable with 0 mean and variance of 1. Okay? So what's going to happen after 100,000 generations? We already know we're going to get some blob, okay? Some very broad distribution. Okay? So I run the code, and this is what we get. Okay? So I run it for many, many times, and I plot the histogram, and this is what we get. Now, if we want control, what do we need to do? We need to add feedback into the problem. What does feedback mean? It means that I'm not just measuring time. If I give you a clock, you have no way to tell what your size is. So we want feedback that you know your size, and based on your size, you grow a bit more or a bit less. Okay, this is the sort of feedback we need. In the same way, like, in the notes that I uploaded to the shared folder, I started by discussing something which has nothing to do with size control. Think about a queue, okay? A queue at a coffee shop or something like that. Let's say there is a rate of people arriving at the queue and a rate of preparing the coffee. Even if the rates are equal, we're going to have a problem. The queue is going to become potentially very long because of these fluctuations. So you need a feedback that if the queue is very long, you either work faster, or probably most likely people won't enter the queue because they'll see a very long queue. So that sort of feedback will keep the queue length confined and not diverging. So how do I add this to the code? Very simple. I add 1% times the cell size, meaning if I'm growing very fast, if I'm too big, then I add 1% with a negative sign to kind of give me a negative feedback to suppress the cell size. And if I run this code, then unlike the mess that we got before, I will get a very nice Gaussian with a tight distribution. The larger I make the feedback, the narrower the distribution will be. Okay? So what we're going to discuss now are stochastic models for cell size control, and also I'll show you how we can use experimental data to find which models are the relevant one, and it's going to start simple, and then it's going to get a bit more complicated as we go along. Okay? Any questions so far? Yes? Okay, very good. So throughout this, unlike next week, where ecology is going to be a lot about interactions, like I mentioned at the beginning of the week, everything here is every cell is for itself, no communication between cells. Okay? And this is indeed the case with these experiments where you keep flowing nutrients, cells are in a constant environment, they don't talk to each other. Of course, in reality, cells do talk and so on, but we have no evidence that that affects cell size control and things like that. Yes?
Where did you add the feedback in the equations?
I didn't. We didn't do the equations with feedback yet. We will. Okay, so that's coming. Just I wanted to give you a hint. We understand now that this timer-based model is a disaster. It's not going to work. I don't need experiments in biology to know it's not going to work. It simply doesn't, right? It doesn't even give you a size distribution, a stationary size distribution. We need feedback. We're going to add the feedback to the equations tomorrow. Okay, other questions? Okay, so in the remaining 10 minutes, I might start with kind of biological motivation to show you the sort of data there is and the sort of models that there are. Okay, so this is, again, a scheme of E. coli. And the sort of questions, what we're going to do is discuss coarse-grained models, simplified models, phenomenological models for size control. We're going to talk about the coupling of DNA replication and division. So we spoke about multiple replication forks, these circular chromosomes, the fact that you have many of them, many replication forks when you're growing fast. And we're going to see that experiments suggest there is tight coupling between these control of division and the replication of DNA. Didn't have to be that way, but that's the way that evolution seems to have shaped this regulation. And maybe at the end we'll have time to talk about possible molecular mechanisms. How is this accomplished at the molecular level? This is still pretty much an open question, but I can tell you about what we know about it. Okay, so one of the models that's been around is that maybe this FTSZ ring, these FTSZ proteins, they accumulate. You're born, a cell is born, it accumulates enough of these FTSZ proteins, and when you get enough copies of FTSZ, it forms this ring around the middle of the cell, and that shrinks the cell into two daughter cells. Okay? So I'm putting it out as a potential model. Note that in this model, DNA replication is not playing any role. So unlike what I just said, we're going to refute this model, but this is a possible model where you control division by accumulating something from birth. In a sense, this is also a molecular mechanism because it tells you this protein is the one you're accumulating and so on. And when you accumulate enough, you divide. Okay? So no coupling to DNA replication in this model. Here is another picture, and it's going to take me a bit of time to explain, but I want to walk you through this. So what do we have here? We spoke already about the fact that you can take fluorescent proteins from jellyfish and stick them into bacteria to tag a protein of interest. So in this case, you're marking a protein that is involved in DNA replication. Okay? And cells are growing from top to bottom, so this is essentially time, only I'm not plotting it as a function of time. I will in a couple of days. But now I'm plotting it as a function of size. So cells here are born at some size, divide at a larger size, and for a given size, the cell, say, starts here and ends here. This is the length of the cell. And I'm plotting the brightness of these fluorescent proteins throughout the cell at a given point in time where it has some given size. What do you see? You see here a bright spot starts and ends here. This is a fluorescent protein that's marking DNA replication. So what does that tell you? It tells you DNA replication started here and ended here. Okay? So in this case, you started duplicating your DNA when you're about 1 micron cubed in volume, and you finished it sometime later. Okay? This is in slow growth conditions. Doubling time was 160 minutes. Now I go to faster growth conditions, and I find that already when I was born, the birth is marked by these dashed lines, the white dashed lines. And by the way, these are averages over many, many experiments. It's not just a single cell I'm showing you. It's the average over many single cells. Okay? Experiments from Johan Elf's group from a couple of years ago. And what you see here is that you started. Why was I born already with replication forks? Because I told you that in these fast growth conditions, you're already born with partly replicated DNA. So DNA replication already started. And when did I start a new round of DNA replication for my grandchildren? I started it here at a volume of 2 micron cubed. Why are there 2 replication forks that I'm starting and not just 1? Because I need to inherit this DNA not for my children, but for my grandchildren. So I need to have twice as many copies of the chromosomes. Clear? And now by the same kind of gist, when I go to even faster growth conditions, I was born already with 2 partly replicated replication forks, and then when my volume was 4 micron cubed, I start 1, 2, 3, 4 replication forks, because that will be for my great-grandchildren. Okay? So this is real data, sort of crazy, and it tells you that cells start duplicating DNA at a volume which is sort of quantized to 1 micron cubed per the number of replication forks. For each replication forks, I want a volume of 1 micron cubed. Okay? So in this picture, you have a critical size not for division, but for duplicating a new round of DNA replication. And division happens when DNA replication finishes, which is a constant time after I started it. So within this model, DNA replication is coupled to division. I do measure size. Like we said, timing is not good enough to control size. I measure size, but I don't measure size for when to divide. I measure size for when to start a new round of DNA replication. DNA replication takes a constant time, about 60 minutes, and then I divide. So that's the picture. Okay? And in fact, this is experimental data from 2016, but there was evidence, indirect evidence for this picture already from the 1960s based on the following observations. Okay, so I'll tell you about that. So the way it's going to work is now I'm telling you kind of a lot of biology, and tomorrow we'll do the more stochastic calculations. Okay, so it takes a bit of time to digest, so I hope you're okay with all this information. Okay. So maybe I'll show you the data tomorrow, but in 1958, there was a nice experiment done where Ilya Schechter and collaborators grew E. coli. It's again, it's like this workhorse of biology, and they grew it in many, many different growth conditions. It's all at 37 degrees Celsius, but they gave it different nutrient conditions. You spoke about poor nutrients, good nutrients, right, with Jacopos, you know all about that. So they grew it in many nutrient conditions. They didn't look at ribosomes concentrations, which you focused on, but they looked at cell size. And what did they found? They found that if you plot the logarithm of cell size as a function of the growth rate, the faster cells are, the bigger they are. Again, a bit reminiscent of what we saw in the Lenski experiments, where cells became bigger as they also grew faster. And there was a linear relation, a nice linear relation between the two. So what does that tell you? It tells you that volume is exponential in the growth rate, that I'm going to call lambda. And the slope gives you a time scale. Okay? That time scale from this plot you found to be about 60 minutes. Is this clear? The observation? I give bacteria good food, they're happy, they're bigger. Okay? Didn't have to be that way. Again, think about the spandrels that we discussed half an hour ago. Some biologists would say, oh, this makes sense because in these faster growth conditions, cells have more ribosomes, and so they need to be bigger to accommodate for more ribosomes. This argument doesn't make any sense. We'll see it at the end of the cell cycle. You could have engineered this in a way that cells would be smaller when they grow faster. There is no inherent, because of what you said before, constant concentration, so size can be big, small, it doesn't matter. You will still have the same concentration of ribosomes, which is the important thing, not the total number. Yes?
But shouldn't there be a saturation somewhere between like the volume and the growth rate? Because
I'm sure there is, but like he took a pretty broad range of growth rate. Some of these are like heart and brain infusion, and some of them are LB media. So he took all sorts of crazy media that the bacteria grow slow and fast. And you see, I'll show you the data tomorrow. It looks like a very nice linear relation. I'm sure it can be broken at some point. Other questions? Okay, so yes.
you said the faster the cells are, the bigger they are, yes?
This is what this tells us.
And by faster, it means they have higher growth rate.
Yes.
It means they have less time for division.
Doubling time is going to be approximately log 2 over the growth rate. So faster growth rate means shorter doubling times. Okay? And a decade later, in 1968, I'll show you again the data tomorrow, people for the first time succeeded in measuring the time from initiation of DNA replication to cell division in E. coli. And they found that for a range of growth conditions from 20 minutes to 60 minutes, this is exactly the conditions of multiple replication forks, this fast regime where cells have multiple replication forks. They found that the time it takes DNA replication is 40 minutes, and the time from finishing DNA replication to cell division is another 20 minutes. So the time from starting initiation of DNA replication to cell division is 60 minutes. Same number. And then Will Donachie understood that you can explain these two observations and why this number 60 minutes appears in both of them in a simple way by postulating the following model. Okay? The model is cells start duplicating their DNA at a critical volume proportional to the number of replication forks, but that's not so important for us. Essentially supported, but what decades later can be measured experimentally using these fluorescent reporters. So I have a critical size for initiation of DNA replication, and then I divide 60 minutes later. Now, for a second, let's forget about these multiple replication forks. Let's assume that it's the simple picture, just to make the calculation simpler. Let's say that my size for duplicating DNA, I call it VC, V critical. So I have some critical volume of 1 micron cubed for duplicating my DNA. And now I grow and divide. When do I divide? 60 minutes later, because from here to division takes 60 minutes. This is what this experiment tells me. What would be then my size at division? My size at division would be this critical volume, e to the lambda times 60 minutes. Okay? Now, if I take a population of cells, size averaged over the population is proportional to size at division, also proportional to size at birth, right? Because that's just a factor of 2. So this would then explain this data. If I have a critical size for duplicating my DNA and a constant delay of 60 minutes, because that's the time it takes to duplicate DNA and then segregate the chromosomes and divide, I would naturally explain what's called the growth law, or you saw other growth laws related with ribosomes, but this is also called the growth law because it's a quantitative mathematical law associated with the growth of E. coli in this case. So you can explain Schechter's growth law based on this simple model. Okay? And I'm going to stop here, but basically by now, we've set up the stage that we have several models on the table, and we need to decide between them. The first model, DNA replication, was uncoupled from division. We accumulate this FTSZ to a critical level and divide. The second model, DNA replication, was triggering division. So division was downstream of DNA replication. And in a third model, that actually Jacopo was involved in setting up together with Marco Lagomarsino, it's both of these that trigger division. So to divide, you have inputs both from cell birth, like accumulation of a protein from cell birth to division, and from DNA replication. So these diagrams for now are schematic. You know, you think of them as schematics, but later this week we will have precise mathematical meaning to these diagrams, where every arrow would correspond to a stochastic equation. You will see what I mean. And we'll be able to use these diagrams to find out which model is the relevant one given experimental data, where, again, we're going to learn from the fluctuations in the data. So that's what we are expected in the next 2 lectures. OK? So see you tomorrow. Thank you.
Now we have the group photo.